Hey everybody, welcome to week 2 of our online field biology, what is this technically called? Environmental Science Lab through Canvas. Welcome! Let's look and see what we're doing this week. This is week 2. I gave some extensions to modules 2 and 3 because I realized I forgot to hit a certain button that made it so you could access it, which is kind of important. So I have decided that I moved those due dates to the end of this week, this upcoming week, instead of today, Sunday. But they'll also, do keep in mind, you still also have these 2 assignments also due this week, okay? So let's talk about these new assignments, modules 4 and 5. Oh, my dog is getting up to something. Oh, my dog is harassing my cat. Hang on. Wait, stop. They're friends, really. They love each other. Okay, if you're wondering, I have a collie mix and a gray cat, and usually they get along, but sometimes the collie gets a little rough. Okay, so what are we talking about this week? We're talking about experimental design. So how do scientists design experiments that kind of follow all the rules? And we'll do a simulation of field work. Okay, so before you do any kind of field work out in the fields, which is literally anywhere that's not in the lab, you have to know what you're doing. You have to think about the safety precautions, because it's different from working in a lab where you're just worried about, oh no, will I get chemical in my eye? Will I, whatever your safety hazards are, do I know where the fire exit is? When you're working out in the field, you have a lot of different things to consider, and it's much harder to get robust experimental results because you can't control for everything, and that's fine. You don't have to control for everything. That nothing in science is ever going to work in a vacuum like that. So we're going to talk about how it's fine that you can't control everything, and that's okay. But starting with experimental design. So this week you're going to these 2 modules. So you've already learned about how Canvas works and how to submit assignments. And if you've had any trouble, you've contacted me. This is also, we talked about how to graph data and how to represent data and how to use sheets, which, y'all, that is literally the tool I use most in any kind of scientific thing. It is just so much. I mean, I either use Excel or Google Sheets, and it does everything. Most of what you need, you can get done mathematically through Excel, and if you can't, you need a higher piece of software anyway, and that's not relevant to this class. Anyway, what else are we going to do this week? We're going to take some of those skills, and we're going to use them to learn the terminology that is appropriate when we're talking about scientific experiments. We're going to design a hypothetical experiment. So hypothetical means just a thinking, it's just a thought. It's a plan. It's not, we're not actually doing it. And then we're going to complete what's called a Labster simulation. So this is a nice way for us to be able to do some simulations online. And so you still get all the content you would normally get in the classroom, but you can do it in your pajamas at home. Okay, so the first thing to know is about when we talk about experimental design in science. An experiment is a test. Anytime you test something out in science, if you're trying to understand the laws of nature, the laws of physics, whatever, if you just want to know if this one fertilizer is going to work best in your garden, you do an experiment, okay? They are a way for us to try and understand how things work. And I used to ask this question, are we even smart enough to understand how things work? Because if you think about it, our brains are just a bunch of cells trying to figure themselves out, and that's very meta and deep and weird. And so it does, the way that the scientific method works is it gives us a framework in order to say, well, we might not understand everything about how things work, but we can at least approach it in a testable manner so that whatever results we get, we can agree they make sense in a certain context, okay? So that's what this is about. So whenever you do an experiment, you start with a question, like, I wonder what this is, or blah, blah, what's this? What's going to happen if I do this? And that in science is called your hypothesis. If you have a whole lot of hypotheses and they agree with each other, that's called a theory. In normal English, a theory is something, I just thought it up, it's just a theory. Because of bad branding, science does not take theory to mean the same thing, which is just a rough part of being a scientist, is having to realize the context in which you're speaking. Some people might not take you seriously if you say theory when you actually mean theory, like the theory of gravity. Well, gravity is kind of a law. If anything can disprove something, that a theory is automatically gotten rid of, but like the theory of evolution, nothing has disproven it yet. And so as soon as it does, scientists are the most excited to get results that say this doesn't fit with your theory, because then they get to find a new theory. But they don't just throw the word theory around randomly, okay? All right, so in these hypotheses, the way we set them up in our experiments is that we're always assuming what's called a null hypothesis, which is that nothing happens, okay? And so basically your end result in how you manipulate your variables, okay, so a variable is something that can change because it is variable. If you control it, that's called the dependent variable. Oh, sorry, no, just kidding. If you control it, that's the independent variable. It's independent of results, okay? So let's say we're looking to see if a new kind of fertilizer will work best in a field, okay? And so if we're going to see if that how much or what kind of fertilizer works best, we need to control most things like how much water, how much light, how much the temperature soil, etc., the temperatures of the soil, etc. But we want to just change the thing we're interested in, that's about our question. Okay, so that's the fertilizer here. And then the dependent is what happens as a result. So if your question in this kind of experiment might be, if I add more fertilizer, will I get more corn? Okay, and so that if I add that thing you're doing, that's the independent variable. The dependent is what happens as a result. Okay. We know, especially with plants, if you got into the whole houseplant craze during COVID, like I did, there are a lot of things that you have to think about when you're taking care of plants or in any experiment. There's always so many things going on, and because we're just testing one variable at a time, a lot of times we try and keep everything exactly the same just so that doesn't confound our results or mess up our results, okay? So our control variables, in the case of this one with the corn, well, I'm testing fertilizer, so I better keep watering the same for all the plants. I better keep the sunlight the same for all the plants. If you do this and you have one plant outside in the sun and one plant in a garage where it's dark, well, that's not going to give you the same results, but might not because of the fertilizer, obviously, right? So you have to, you really only test one thing at a time. Your control group is going to be the one where nothing it changes, okay? So it's either your baseline you're comparing everything to, or it's the one that gets no treatment. The experimental one is the one where you're actually doing stuff to it, okay? So in my control group, I add no fertilizer. In my first experimental group, maybe I add 5 grams of fertilizer. In my second experimental group, maybe I'll add 10, okay? And then you want to do, if you can, you always want to replicate this, because if it happens once, who knows if that's a real result. Or if it's just something random. If it happens twice, still could be random. 3 times, you're getting better, but the more you can replicate something, the more likely it is to be true. Okay, so in science, we have this big problem right now with people not getting enough funding to replicate experiments, and so people it's a problem with our dopamine systems, how everyone just really wants to know the new hot thing right now, and they forget to go back and look at the thing that actually works. And so that's one problem in science right now, is just getting people to repeat experiments, to look and see if you replicate it, do you still get the same results? Because then that suggests very highly that your experiment worked or is valid. Or do you get totally different results and then there's something wrong with your experiment altogether and you have to go back and think about it, okay? So replication or repeated trials, very important. Oh yeah, so treatments. If you want to go ahead and add a second independent variable, like the amount of sunlight, you might have treatment groups within your experimental groups, okay? So that could get a little more complicated. It also helps you kind of conserve resources a bit better, especially in the case of doing something outside where you don't want to disrupt nature as much as possible, but you do want to get good results. All right. Cool. Why do we need to know all this? Oh, because now it's time for a quiz. So take a quick quiz where you are a medical researcher developing a weight loss drug, and you need to, okay, so your rats that took the drug were fine, but the autopsies show some heart valve damage. So how do you determine if it's your drug causing the heart valve damage or if it is something else? I will tell you, I didn't write this assignment because the idea of a weight loss drug is gross to me. Love yourself. You're all beautiful and wonderful. But you can still design an experiment around this to show, like, well, is it the drug doing it or is it something else doing it? So what I would like you to do is go ahead and answer this quiz, okay? Then once you're done with the quiz, you're going to have a chance to, so you're going to, let's see. Yeah, you're going to continue, sorry, I'm rereading this for the first time in a while, so it's fun for everyone. You're going to design your experiment based on what you did in the quiz part. And then you're going to do the Labster demo. And the Labster demo is doing something called electrophoresis, which is how we separate out different genes. You may have seen this on like different kinds of, like CSI. They always show these things because they're like, oh my God, look at the electrophoresis. Oh, yeah, I just get my DNA to tell me who, blah, blah. Yeah, so this is what electrophoresis ends up looking like. It's where you have a gel. This is it's called agar, which is basically vegetarian jello. And you run DNA samples through it to see how big they are, okay? And we can do this to see if they match something that we found on a crime scene, or if you are looking at different animals to see how they respond to certain things. Do they make the same protein? Whatever. This is what electrogelphoresis looks like. This is what a picture of it looks like. Anyway, in your demo, you're going to be looking at this and having you play with some experimental some molecular design, I guess, is what I want to say. And you might say, I am in this class because I'm an environmental scientist. I don't do molecular science, but unfortunately molecular science is everywhere. And one of the best ways to understand what's happening at the big level is to understand also what's happening at the micro level, which is the DNA level. So don't worry, it's all good. So you're going to load this in the window itself, and it's going to walk you through how to set up this lab, how to do the lab. Okay, then there is another one you'll also do. Okay, cool. You got 2 labs to do, a quiz and an experimental design. Here's some extra resource if you want to listen to another guy talk about it. And then just reflect. And as you know, once you reflect, you are done with that module. So that's all you have to do for the experimental design module. Next up is the fieldwork simulations overview, okay? So, again, this is an environmental science class, so why are we inside? We should be outside all the time. Well, but we should be learning how to be proper scientists outside before we actually get into the field and start doing these things, okay? You're going to be doing another labster. Oh yeah, and the designs. Okay, so let me tell you, the money for computer graphics and design is not in science research. It's obviously in the big gaming companies. So you will see that the games we play in Labster are not as pretty, but they still give you the good results, and that's why we use them. Okay, so you're going to be looking at 2 big things. You're going to be looking at biodiversity, which is, if you look at, this is actually something we deal with a lot when we're talking about agriculture in the U.S., because we have a bad habit of just having monocultures, which is just one huge field of corn. Cool, I'm never gonna do anything else but corn. Oh, it turns out you can't do that, because then that one organism will use up all of its nutrients in the soil, and then you have to amend the soil. Or it will, by decreasing, by only having one species of something in a field, it can be wiped out by a virus very quickly, okay, because it's all the same thing, and so it'll all get sick the same way, and then, oh no, it's dead. Oh, and so you're gonna learn why biodiversity is so important, okay, and remember we have that textbook that if you need to go back and look up some terms. And you're going to be doing this through an exoplanet. How exciting. Love aliens. Okay. And you're going to learn about the biodiversity index, which is sometimes called the Shannon index. And you're going to learn when do you prioritize sampling. And then once something goes very nicely hand in hand with looking at biodiversity of an area is the spatial ecology. And this is the stuff I love so much, where you can look at, areas and see, well, I know only this one kind of bird will eat at this level, but then another bird eats at this level, and then another bird eats at this level, and sometimes they'll interact at the barrier, or at the interfaces, and they might like have a squabble or whatever, but often they stay in their specific niches and they don't overlap. Okay, so when you look at how ecology works in different spatial spaces, it gets really interesting, because you can have bugs that maybe they never understand that the other one exists because they just don't ever go into that region of space. There's also something called temporal ecology, or temporal speciation, which is where in frogs, certain species of frogs, they don't hear each other's calls unless they're in mating season, and so they could be sitting right next to one frog that's actually pretty closely related to it, and that frog is screaming its little heart out, oh my god, I'm ready for frog sexy time, and the other frog who's not in mating season won't even hear them because their brains aren't primed and ready to hear, sexy frog time sounds, and that's actually separated them into different species as a result. So that's fun. Okay, so you will do this again on the Labster. Fun, fun, fun. It's going to teach you things like quadrat, camera trap. Camera traps, you're seeing a lot of these now, especially with, we're learning a lot more about the biodiversity of California and the U.S. in general because people are putting wildlife cameras out just like on their properties or in forests or like parks are doing it. And all it does is it just takes a picture as movement happens. And so you start to see interesting things like recently there was a report out in Los Angeles of a fox and a badger working together. I didn't even know I had badgers in Los Angeles, y'all. No idea. You'll look at pitfall traps, which is something we use for bugs mostly and lizards. It's literally just you fall in it, okay? We don't use that for bigger animals because oftentimes it causes harm to the animal, like breaking the leg, so we don't do it to big ones. And then you will look at elevation biodiversity gradients. That's very interesting. As you go up a mountain, you see the same distribution in your plant life and then following that the animal life as you do as when you're moving away from a beach or a place with lots of water. So it's very interesting. Okay, so you're gonna do this one and then another one with spatial ecology. 2 little labs. Reflect on it. Tell me what you thought about it. Did you learn anything? Did you like this? Did you hate that? Whatever. Do you want to know more about spatial ecology? Do you want to know more about biodiversity? Whatever about it that you wanted to reflect on. And then as you know, that means you're done with the module. Okay, so that's what you're doing for this week. You're going to finish up simulations 903. 2 and 3 from last week, the graphing ones. Then you're going to do your experimental design Labster and the quiz and the design experiment based on that. And then finally do the fieldwork Labsters as well. Have a good week. Please let me know if you have any questions. I'm totally here for you. And be fabulous.